In this video lecture, I'll be talking on live attenuated vaccines. And as you may know that vaccination is a prevention of disease by vaccine. And live attenuated vaccines are artificial vaccines. Actually, they are artificial active vaccines. In live attenuated vaccine, the pathogen get attenuated, as can be seen here, attenuated, and then delivered to the body. The immune system starts to deal with it. Following that, antigen present in general, not only macrophages, but antigen presenting cells, these are the dendritic cell and the macrophages, engulf the attenuated pathogen, process it into peptides which bind to the major histocompatibility class 2 as the rough endoplasmic reticulum. So peptides together with major histocompatibility class 2 will be expressed on the surface of antigen presenting cells, whether it is dendritic cell or macrophages. So T helper 1 will interact with the peptides and with the major histocompatibility class 2 through the T cell receptors and the CD4 protein on the surface and get activated. Activated T helper 1 produce interleukin number 2, which in turn activate B cell and T cells. This here appear very clearly. So this is the B cell. A B cell also, as an antigen presenting cell, has the capacity to internalize the attenuated or the live attenuated vaccine by means of endocytosis, and this presented in association with major histocompatibility class 2 to T helper 2, and T helper 2 here get activated and produce some cytokines, for example interleukin number 4, which is growth factor for B lymphocytes, which get activated and differentiate into memory B cells and plasma cells. Memory B cell remains in the body and recognize the pathogen if the body encounter it again. I mean, in the future or during the future infection. Plasma cell produce antibodies. These antibodies protect against pathogens, and if the pathogen affects the individual in the future. So this according to longevity or longevity theory, that plasma cells, they can also produce together with antibodies together with memory cells that has the capacity to memorize the pathogen. So these are examples of live or some examples of live attenuated vaccine. For example, Bacillus calmette-guerin, which is attenuated Mycobacterium bovis and protects against human tuberculosis. And also other examples are the measles, mumps and rubella MMR vaccine, and the varicella chickenpox vaccine, or oral polio vaccine, Sabin vaccine given to children, and yellow fever vaccine. However, live attenuated vaccines may not be suitable for immunocompromised individuals. For example, patients with debilitating diseases like tuberculosis, or patients with cancers, or patients with autoimmune disorders, as the pathogen may regain its virulency and may lead to a disease. So live attenuated vaccines stimulate both cellular and humoral immune responses. In contrast, the inactivated vaccines stimulate only antibody immune responses and no cellular immune responses. And as these are some examples of inactivated uh, vaccines, for example, inactivated polio virus vaccine, whole cell pertussis vaccine, rabies vaccine, hepatitis A vaccine, tetanus, and diphtheria toxoid vaccines. Thank you very much, Professor Ahmed Bolad.